Putting this talk together was a bit of a challenge. Most of the talks that I give are sort of basic science oriented, and so trying to get the right information for this audience in terms of what would really appeal to you in terms of the projects we're doing, while at the same time sort of talking about the genesis of the project is what I hope to accomplish. So I would love any feedback if you feel like I hit the mark or missed the mark, and why you thought so. I would appreciate it. All right, so title is Hepatitis C Vertical Transmission, Call to Egypt in My Journey into Global Health. So away we go. All right, so first some acknowledgments about people in my lab or research colleagues that have made much of my research over the last several years possible, some of whom are in the room, like Shelton. And then obviously a big acknowledgement to the DGHI, which has really helped me kickstart this project, the reason why I'm here. So I appreciate the opportunity that they've given me to really move this forward. All right, so objectives of the talk. I'm going to give you a basic overview of hepatitis C virus, a little bit about what is known about vertical transmission, and then talk about some of our work in the lab, modeling this process in vitro and some of our preliminary studies. Then I'm going to talk about Egypt and their unique hepatitis C problem, and then describe what plans we have for studies there related to this topic. One figure about the virus. Hepatitis C virus is what's called a plus-sense RNA virus. For those of you that don't study viruses routinely, that means that hepatitis C can get into a cell and then immediately start making proteins to get to work. This is what its genome looks like. There's what are called structural proteins that make up the key structural parts of the virus. And then the non-structural proteins that are really responsible for different aspects of replicating the virus, making new virus. As far as clinical features, you're probably familiar with some aspects of this. Hepatitis C, one of the unique characteristics is there's a high rate of progression to chronic infection. So most adults, when they get infected, about 80% or so, end up with some sort of form of chronic infection. That doesn't mean they all have symptoms of liver disease, but they have evidence of continued viral replication. What's interesting is that children are different. They seem to resolve their acute infection at a higher rate, so their progression to chronic infection is lower. We don't exactly know why this is, and probably a lot of it is developmental, and that's another aspect of my research that I'm not going to talk about today. So natural history of progression in chronic infection. Some patients may develop cirrhosis, which is a overwhelming fibrosis of the liver to the point where it can't function anymore. End-stage liver failure is basically when you're left with no functioning liver cells. And then hepatocellular carcinoma is out of this fibrosis, you run the risk of developing a liver cancer. How do you get hepatitis C? The major routes are if you were and I'm talking about the United States now. If you were transfused prior to about July 1992, that time frame has to do with the reliability of our testing for the virus. Prior to that, most of the tests relied on antibody tests that didn't pick up all of the hepatitis C proteins. And so by 1992, we had testing that was sophisticated enough to reliably detect virus in infected patients. If you also received any blood or tissue, so if you had a transplant, if you had a bone graft, if you had something else, prior to that time, you could have been infected. Certainly the most prevalent way that people acquire the virus now, particularly adults, is through IV drug use, contaminated needles, or contaminated drug paraphernalia. Okay? Vertical transmission is the primary route that kids get the virus. This is passed on from mothers to their infants. The timing of this is something we'll talk about. It's not clear when this happens, whether it's at delivery or prior to delivery. I'll show you some information that makes me think that it's prior to delivery. And then the other risk factors are sexual transmission. Sexual transmission probably does happen, although at a very, very low rate. And it's not clear that it occurs in the absence of some minute blood exposure that's associated with that, whatever type of sexual activity you're talking about. We know that, for instance, men who have sex with men, where perhaps the sex is more traumatic, that there's a higher risk of transmission in that population. And then we're still left with patients who have hepatitis C that's diagnosed, and they swear up and down they don't have any of these risk factors. And so the question is, are there other perhaps minute blood exposures that are out there? So whether you use a razor that has some minute blood, or toothbrushes, there are other potential minute exposures that may be transmission. Again, not frequent, but possible. And that's yet to be defined. And I think there's also some aspect of people that may not necessarily be telling the truth when they say they have no risk factors. So just a little bit about impact in children. We think that there's somewhere between 20,000 and 50,000 kids that have chronic HCV in the U.S. And we did some studies that sort of estimated the cost here between 170 to 400 million. So in kids, this can actually be a significant problem. And so while there have been a lot of programs to halt bloodborne transmission, approximately 1,000 kids per year still acquire HCV by vertical transmission. And this number is based on the final sort of chronic infection, and we'll talk a little bit about that. So that makes this the leading route of acquisition in kids. Cirrhosis in kids, while infrequent, does happen, and the longer you're infected, the higher the rate. And so it's possible that we may see some of these, the rates of cirrhosis go up in the coming years. And there's currently no vaccine and no safe drug that halts vertical transmission. There is now one large-scale clinical trial that was completed. I apologize for this not being totally up to date, looking at antiviral therapy in kids. But it's taken a long time to get to this point. All right, so let's focus on vertical transmission now. So what do we know about vertical transmission with Hep C? So we know that the transmission rate compared to other viruses is actually pretty low. Somewhere between 5 to 10% of all infants have the virus transmitted from their moms. However, many of those infants, probably about half, will resolve that infection somewhere in the first 2 to 3 years of life. It's not clear why this happens. Again, it's probably developmental, probably immune-related. It's the combination of them that makes them different, infants different than adults. But anyway, so at the end of, let's say, 3 years, about 3 to 5% of these infants have chronic Hep C infection. In the pre-treatment or highly effective treatment, HIV co-infection was a huge risk factor. So between 20 and 25% of infants born to co-infected moms ended up with Hep C. While there haven't been large studies to show it, our sense is that now the current rate is about the same as mono-infected patients, if your HIV is controlled. Okay? Studies have been shown looking at C-section. So C-section for viruses that are transmitted in and around delivery have been effective in reducing transmission of many viruses, but the studies looking at C-section with hepatitis C have shown there's really no difference in the rate of transmission. So I would say that this argues against transmission being a delivery event, and really that transmission happens earlier in utero. Okay? And studies, sort of very simple studies, have looked at things like amniotic fluid. So we don't think that hepatitis C is sort of an ascending infection. The infant isn't being bathed in virus that they somehow consume. But there really haven't been other detailed molecular studies looking for a virus beyond that. So the rationale for the project when we started this was that we needed to understand the determinants for vertical transmission in order to enable some sort of prevention. Most countries in the world aren't able to afford the standard of care that's present in the United States. This is treatment that costs somewhere between $20,000 and $40,000 for a year's worth of therapy, and most countries just don't have those kind of resources. So let's just review a little bit about placental anatomy. If you're talking about a virus that's transmitted in the placenta, basically, to make this very simple, you're moving from the mother side here over to the fetus or infant side. And to cross over from the mother's uterus into the baby, you have to cross this middle layer, which is called the trophoblastic layer. Okay? The trophoblastic layer is really what forms the barrier between mother and infant. This layer is made up of two types of cells, the syncytiotrophoblasts and the cytotrophoblasts. The differences between them are not really relevant for this talk at this point. But basically, they really form the filter. And for something to pass through, they have to deal with this layer. So what would it take for vertical transmission to happen? So it has to, as we talked about, it has to cross from the mother to infant side. If you're talking about in utero infection, basically the virus has to somehow infect this layer, whether it's the syncytiotrophoblasts or the cytotrophoblasts. And these cells would have to support some level of infection. Okay? One would assume, based on the rates being very low, that they're not nearly as efficient at being infected as liver cells are. Okay? So that's the rationale we're starting with. So our hypothesis is that there are significant differences between liver cells and these cytotrophoblasts that affect the regulation of the virus. Okay? And this is how we started out with this project. So I'm going to go through a little bit of what we've done in the lab But just want to review our approach for those of you that aren't molecular virologists. What does it take for a virus to infect a cell? So first, the virus has to actually bind to cell surface receptors on the cell. Okay. Once it does that, it enters the cell and then finds its intracellular target. So some viruses replicate in the cytoplasm of the cell, some in the nucleus, but each sort of has its particular target that it needs to find after getting in. Once it does that, the virus needs to make its own proteins. Viruses don't carry luggage. Okay, they use the host proteins, and so they need to identify its host factors and then get going. So they usually usurp many host factors to do this. Okay. Once the virus has done this, it assembles these new proteins and nucleic acids into new virions, and then ultimately these virions may exit the cell in order to affect other cells. Sometimes this actually kills the cell, sometimes it doesn't. In the case of hepatitis C is not cytolytic, which means it doesn't kill cells. All right, so what do we know about hepatitis C and some of these requirements? So almost all of the studies have been done in liver cells. We know that many of the receptors are co-receptors here on liver cells. There are several different proteins that have been identified. There have been no studies that have been done in placental cells. In terms of critical host factors for replication, we know that a microRNA called miR-122 that's enriched in liver cells is clearly very important for the replication of the virus. And no one knows anything about these in placental cells. As far as host factors for assembly and release, one step that's a key step in the liver cells is the binding to lipid droplets. So there are little fat collections within the cells. One of the hepatitis C proteins, the core protein, binds to these droplets and really forms a scaffold by which the rest of the virus can assemble. Okay? We don't know anything about what happens in placental cells. Okay, so this is a huge unknown here that we're trying to tackle. So the strategy is we try to look at these key steps, translating them from what's been done in liver cells, and then start to look in placental cells. So the easiest way to do this to start is to model this in the laboratory using cells in a tissue culture dish. So the cells we use, the liver cells, are called HUH7.5 cells. These have been used for about the last 15 years or so to really study the hepatitis C life cycle in detail. The cells we're using as our placental or trophoblast cell line is something called HTR8 cells. These were cells that were isolated from a human placenta from the first trimester. They were immortalized with an unrelated viral protein, so they can essentially grow indefinitely. This is now almost 20 years ago that these cells were developed, and so they've been replicating and reproducing for the last 20 years, propagated throughout labs around the world. Okay, so this is our model system. The reason why we chose these is in addition to them being immortalized from a hormone and protein standpoint, they very much resemble primary isolated cells. So if you were to look at trophoblasts isolated from a first trimester placenta, these behave very much like those do. Okay? So first we started with analyzing cell surface proteins. So the virus has to actually get into the cell. If you don't have the right cell surface proteins, the virus is not going to get in. And so we've performed simple flow cytometry experiments, which is basically using antibodies to detect what proteins are on the surface of the cell. We looked at the receptors that I told you about that hepatitis C uses. And basically I'm going to show you two flow cytometry studies, just basically any shift of these curves to the right here, this way, means there's more protein being expressed. So these are our liver cells here, and for the LDL receptor and the scavenger receptor B1 and Claudin-1, these are the liver cells which clearly we know express the proteins. These are our placental cells that also express some level of protein. Some higher, some lower, but the fact is that they're on the cell surface. When you look at some of the others, we notice that there might be one discrepancy here. CD81 is sort of the primary receptor for hepatitis C. There's lots of CD81 here with a massive shift of this curve to the right. There's really very little occludin here compared to sort of our background controls. So there might be this little blip here. So that's interesting. And keeping in mind that these cells are first trimester trophoblasts, I went back and looked for some information about whether this was known. And in fact, it is known that occludin, this protein, is not expressed at high levels during the first trimester, but actually goes up as you get later in pregnancy. These are what's called Western blots that look at the intensity of expression, and this is basically just the darker the band, the more the protein is present. And so if there's not much occludin early on, and then more occludin later on, perhaps it means that hepatitis C doesn't cross early in pregnancy but at a later stage, maybe in the second or third trimester as occludin levels go up. Okay, so that's been interesting for us to find out. The other thing we've been doing, and this relates to some previous studies we've done, is looking at lipid droplets in these cells. So what we did is we took some of our hepatitis C proteins, we put them in these trophoblastic cells, and we wanted to see how they behaved with relationship to these lipid droplets. And we used various methods in the lab to identify the proteins and look at the lipids. And basically what you'll see is on the top set of panels here are liver cell lines we use in the lab. And what's nice about these cells is that if you look at the pattern of this fluorescent green staining, that's where the hepatitis C proteins are. And then if you look at the pattern in the cells here when we stain for the lipid droplets, there's almost complete co-localization here. So actually if this picture was a little more high resolution, you'd actually see little rings of staining around the droplets. Okay? And that's what we expect in liver cells. That's how the protein behaves when you put it in a liver cell. What's interesting is when we looked at these placental cells, what we saw is this big clump of green staining. Okay? And it looked like the protein was sort of stuck in one place in the cell. And it didn't really translate with the nice ring-enhancing lipid. And actually you can see some areas of sort of hollow staining, which is actually where the lipid droplets are. So it doesn't really correlate in the same way where the proteins are finding on lipid droplets. So again, these are differences that may be relevant, but it's hard to know at this point. We know that the protein, in order to travel lipid droplets, needs to be processed by a cell. This is a particular enzyme. If the protein isn't cleaved, it could be because you don't have the enzyme to cleave it, or perhaps because the enzyme doesn't work in these cells in the same way. When we looked at these cells, we basically found that, using the same Western blot, that there wasn't much of this protein around in these placental cells compared to the liver cells. And so it's interesting to know that many of this protein has some of its normal host substrates for cleavage are immune-related proteins. So if you think about the fact that pregnancy is an immune-privileged event, your immune system reacts differently when you're pregnant because you don't want to react against your growing fetus. And so it makes sense that in a placental cell, this protein might be at lower levels in order to change the immune processing of certain molecules. So again, this is very interesting and potentially could be significant. So we haven't yet looked at this microRNA. It's on our list of things to do. And what we also decided to do was see if we could actually infect these cells in the lab. So there are strains of hepatitis C that we can use in the lab. One is called this JFH isolate that allow you to actually look at whether a cell can be infected. So based on what we saw, we didn't think that this would happen at a very high level, but we decided to try. The panels on the left here show our control liver cells, which the big bars basically show the amount of viral products that we can measure. If you put a lot of virus or a little bit of virus, you get the resulting amount of viral RNA. If you do this in the placental cells, you get some increase in signal, but it's hard to know if this is really robust because this is a log scale. Okay, so these are tenfold differences as you get higher up. So this difference is pretty piddly. So it's hard to know how much replication is actually happening in these cells. So at this point, if you're an academics, what do? You try to write a grant. So we wrote a grant to the NIH describing our finding and saying we think this is a model for hepatitis C. The reviewers were not so enthusiastic. And basically what they said was that this model may not have any connection to what's happening in patients. That basically you're studying cells in a dish, and what you found may only be relevant to cells in a dish. And so the need to actually identify patient samples to try to do some of these studies to investigate what was happening with hepatitis C became a real priority for me. Okay? So if you were to try to do a study of vertical transmission in the United States, at Duke we see about 5 hepatitis C infected pregnant women each year. Okay? It's very low. Partly because there aren't a whole lot there, and partly because of just where we are and the fact that Duke is only one of many providers. Okay, so let's say you wanted to get a significant number of patients for a large trial. So you need probably 10 or 12 U.S.-based centers. You'd have to screen somewhere to get to about 50 or 100 women per year. Maybe you'd be lucky and half of them would consent since you're only collecting their placental afterbirth, which otherwise gets thrown away. And if you had, at best, a transmission rate of 10%, it would probably take you 4 to 5 years to collect tissue from 50 women. Okay? And you can imagine 5 years, 10 to 12 centers, screening these patients. This is major bucks. Okay? A very difficult trial to do. And again, I'm giving you best case numbers here. Okay? So, perhaps there are alternate strategies. So, where else could you do this study? And the sort of the question that goes with that is, what country has the highest rate of hepatitis C in the world? And the answer to that question is Egypt. Okay? And let's talk a little bit about Egypt and why hepatitis C is so prevalent there. So, in the early 90s, when our testing for hepatitis C became more reliable, studies being done in Egypt showed really very alarming results in terms of the prevalence of hepatitis C there. This was from a large study that was done in a town, a city sort of area north of Cairo. They found that 12% of rural school children tested positive for hepatitis C, 18% of rural inhabitants, 22% of army recruits, 16.4% of kids that had enlarged liver or spleen, almost 55% of kids with multiple transfusions, 47% of adults with chronic liver disease or cancer. The other thing that they found was that the seroprevalence was largely age-dependent. So if you looked at kids under 5, there was almost no hepatitis C. Whereas if you looked at people over 50, there was 40%. Okay, so massive amounts the older you got. So clearly there appeared to be some age-related exposure that was at work here. The major risk factors when they did larger studies related to history of transfusion, which obviously makes a lot of sense if you don't have a safe blood supply and you couldn't reliably test people. If you had a mother with hepatitis C, which we know for a vertically transmitted virus makes a lot of sense. But the other thing that really caught people by surprise was history of being treated for schistosomiasis with an IV tartar medic. So schistosomiasis is a parasitic organism. It lives in fresh water and is carried mostly by snails. It can infect either your liver or your urinary tract, depending on what species it is. And it's endemic in Egypt, or was endemic in Egypt. Some of the history is that one of the treatments was this IV tartar emetic. Okay? If you looked at the newer agents as time went on, some oral agents, oral praziquantel is also used, that the patients that were treated orally with the praziquantel did not have increased rates of hepatitis C. So it really pointed at this IV treatment as really being the key factor in infecting patients. So if you went back to sort of the evolution of this process, in the 60s, the Egyptian Ministry of Health decided that given the high burden of schistosomiasis they had and the resulting liver disease, they would initiate some preemptive therapy for patients. And so in rural areas that had really high levels of the schistosomiasis parasite, they would screen patients looking for the parasitic eggs in either their urine or their stool. And if patients were positive, they put them in this treatment program where they got 10 to 12 doses of IV tartar emetic. Okay? And this program ran from the early 60s until the mid-80s when an oral agent became available. So if you think about this time frame, this is our blood supply was not safe. We didn't have disposable needles, so they were reusing supplies with perhaps some hasty sterilization techniques, if at all. Okay, and so this is really the key risk factor that makes Egypt unique. This just gives you an idea about this age cohort and how there were not many young people infected, and that this population here that was the target of that treatment program is where all the hepatitis C was. Okay. If you look at the current seroprevalence in Egypt now, it's about 10 to 15%. In the United States, it's about 1 to 2%. Probably about 2%. The major caveat with doing studies of hepatitis C in Egypt is they have a different genotype than we do. There are 6 genotypes of hepatitis C. They have genotype 4. Our genotypes are either 1A or 1B, the majority of them. Okay, so that might be a potential difference in terms of looking at hepatitis C is, do the genotypes behave differently? I can tell you that there's been at least one large study of vertical transmission in Egypt that show essentially the same patterns we see in the US and Europe. So my suspicion is that that's really, the genotype differences are not going to be that significant. So if you reran the same study numbers that I told you about, doing a study on vertical transmission in Egypt, if you have a 10 to 15% seroprevalence rate, you could identify potentially 300 mothers a year, depending on what hospital you were at. Let's say 30 of these would transmit virus to their infants. Perhaps half or more would allow placental tissue analysis. So you could do this kind of analysis at one moderately busy center. So I did this math based on 3,000 deliveries. The reality is a busy hospital in Egypt does way more than that. And that would be just 1 year. Okay? And then if you think about the fact that study costs in Egypt are much less than here. So this becomes very feasible in terms of doing this study in a way that doing it in the US is not. All right, so in order to do studies in Egypt, since I didn't know anyone in Egypt and had never been there, or at least been there for like 2 days when I was 14, I wanted to try to contact some of the people who had actually done work there. So many of the studies that I cited identifying the schistosomiasis as a major risk factor were done at the University of Maryland or based there by someone named Tom Strickland. Okay, so I contacted him directly about initiating some studies and seeing if he had some active protocols still there. So he had sort of semi-retired, but put me in touch with someone named Summer El-Kamari, who was sort of his junior faculty member, originally from Egypt, but trained in the US, that had an interest in hepatitis C and was now running this network from the University of Maryland. Okay? So we arranged to meet, to talk on the phone, and really hit it off immediately. Our expertise and interests were very common, but our skills and interests were actually quite complementary. He was more interested in sort of the epidemiology and immune mechanisms related to vertical transmission, whereas I was more interested in some of the basic science and molecular virology. So it really became like, how can we work together to make this a working program that really could shed some light on what was happening. So really the key step was the DGHI support at this time. So the announcement for sea travel happened pretty much right as this phone call happened. And so I was able to write a proposal to actually visit investigators in Cairo along with Summer and meet the key people that might help us do this study. Okay, so the grant was awarded back in October of last year. We had originally planned the trip for March, but you can imagine, given the political events of late January and early February, we decided that March probably wasn't a good time to go. So we ended up going in July, in mid-July. The other thing is actually we were able to line up some other support. So Merck has what's called an investigator-initiated studies program where investigators can submit proposals on related topics for them. We proposed this same pilot study to examine placental tissue from HCV-infected in women. Normally, this would not really be on many drug companies' radars. The appeal for Merck was that they are one of the 2 companies that now have a newly approved agent that directly targets the virus. Most of the other treatments against hepatitis C are really modulate or enhance host immune responses. But there are 2 new drugs that target one of the viral proteins. And so Merck is one of the companies, and so this became something that was potentially interesting to them. And so they have given us a year's worth of funding. So by the time we were going to Egypt, we actually had some money in hand saying, we want to do this project, and we actually have some money. So it really became very appealing for them to want to get involved. So things worked out pretty well. All right, so what did we accomplish during the trip? So it was sort of a very compressed agenda, but I think we managed to really hit the highlights. So basically 2 full days of meeting with people there at Cairo University, which is a large academic center at Cairo. We met at one meeting with the head of OBGYN, the head of outpatient services, a public health representative, the head of pediatric GI, the head of the neonatal ICU. So actually, the local person there was actually able to collect all these people into one place so we could speak to everybody together, get them on board with what we were doing. So I was really amazed because I think you can imagine if we tried to do this here, it would take us 6 months to schedule this meeting. Okay? We all know that. So the fact that with maybe a month's worth of lead time, our local person in Cairo got all of these people on board. One, it, I think, speaks to the fact that they are really desperate to do something about this problem, and that there's enthusiasm on board for doing this kind of research. Okay? So the fact that we were speaking to a receptive audience, I think, also is really important. The other key thing is that in order to examine placental tissue, we needed to have someone from pathology really on board to talk about how to analyze the tissue, how to examine the tissue for the virus, what kind of techniques that we could do that haven't been done before. So we met with the chief of pathology. He basically runs the referral lab there, and they have access to the latest techniques there. We basically discussed this is how we envision this project. This is how we envision utilizing these techniques locally. We would develop the protocols here, and then we would basically give you these protocols, and you can use the reagents and hopefully do all the work there. One of the things that we realized early on was not going to be allowed was getting tissue from there. They were not going to send tissue out of the country. And so that was something that was made clear to us pretty early on, and so we knew that what we were doing was developing protocols here and then having local people do it, which I think also it helps them because they're getting expertise and knowledge out of it. Okay, so some of these are, I'm trying to translate some of the lessons that I learned while doing this, is that really it needs to be mutually beneficial. You can't just go and say, we're going to get this from you. They really have to feel like they're getting something out of it. Okay? The other thing we did is we met with many of the local investigators from the Maryland team. So the Maryland team has a group of people who are on the ground in Cairo. There's one person who's trained in pediatric infectious disease who's there as sort of the coordinator of all of their studies. They have several researchers, a woman who has a PhD in immunology, and they have all the local facilities that they've sort of co-opted from local Egyptian groups to basically process all the samples for storage and the like. So we knew that we had all the infrastructure locally to be able to do the study we wanted to do. So the proposed study design that we have in mind that we discussed was active screening of pregnant women for hepatitis C RNA. We know that you have to have chronic infection in order to transmit, so you need to confirm that these women are chronically infected. You're going to approach those that test positive for consent. You're going to then collect the placental tissue at birth. Okay, and the way we're going to process the tissue is really we wanted to do it in parallel. Okay, so there's a couple of methods we're going to use. One is flash freezing and something called OCT. This is a special reagent that allows for preservation of both tissue proteins and tissue nucleic acids. This is critical if you actually want to look for the virus. This is, while it's not incredibly expensive, it's not what's usually used. What pathologists usually use when they process tissue is basically classic paraffin-embedded tissue. So you take like what's essentially a wax, you stick the tissue in there, and then you're able to cut thin slices and analyze them in the lab. But the problem is that paraffin doesn't preserve all of the proteins and RNA that you would necessarily want routinely. You'll have some, but you may not have them all. So the idea is if we process tissues both ways, we adapt our methods for our research protocols, and then see if once we identify tissues that are positive, if we can go back to the paraffin tissue, then the protocols become generalizable. Any pathology lab that's processing paraffin-embedded tissue could actually look for the virus if they wanted to. So that's the idea for doing it in parallel. The other thing we need to do is actually follow the infants at somewhere between 3 and 6 months to see if they actually were the targets of transmission. So did the mom actually transmit the virus? Just because the mom had HCV doesn't mean that the infants are going to get it, right? So you have to confirm who actually transmitted so you know which samples to actually go back and look at. And so once you have those, you then can go back and batch test all of these samples looking for hepatitis C RNA and some of the proteins. We have a new in situ hybridization that basically looks for the nucleic acids and uses a label to show you which cells have these nucleic acids. You also have what's called immunofluorescence, which was those fluorescent green pictures that I showed you looking at any of the key hepatitis C proteins. They have to be there in enough of a quantity to give you a signal, but these are the best methods we have right now. The other thing you obviously need to do is we need to find longer-term support. We only had a year's worth of funding, which was going to let us see some patients, but wasn't going to be enough to really do a full-scale study. So we were lucky in that the NIH just had an announcement for placental infection, so we wrote a proposal to them to really do both the patient study and some of the in vitro work. We also found that USAID has a program called the U.S. Egypt Science and Technology Grant. This is basically to build not just medical scientific programs, but any science-related program which is collaborative in nature, involves both U.S. and Egypt institutions, and also is aimed at really building local infrastructure. And so what we've done is say to them, we are helping create this research protocol here to look at this problem. We are going to export these methods to local investigators in Cairo. We are going to also, there's a training component here, so actually junior investigators or trainees from Cairo are going to come and spend a month in my lab at Duke learning the methods here so that they could potentially adapt them to other pathogens if they're interested in herpes, or they're interested in chlamydia, or they're interested in something else. We could potentially use some of these molecular techniques to look at the way that the virus is transmitted. And so these were both just submitted within the last month, so we won't hear about them for several months, but the idea that we're actively seeking support, I think, is also reassuring to the Egyptian investigators that our interest is really more long-term. And then the other thing we need to do is just refine our methods. So I just wanted to show you a picture about some of the new methods we're using. So this is a new in situ hybridization method. So hepatitis C has been notoriously difficult to actually find in tissue. And we're using in vitro infected cells where we take a virus and then infect cells. And perhaps looking at the TV screens is better than looking at the pictures are better in terms of the quality. But on the left-hand column are uninfected cells, so there's no red signal there. In the middle are cells infected with a lower amount of virus. And you see perhaps one lone cell that's infected at a somewhat low level. And then on the right-hand panel, you see cells infected at a higher level. And you see that there are multiple cells together that are infected, and there's perhaps more signal in those cells. So the idea is that we're going to take these methods that we use in the lab and then translate them into actual patient tissue to try to see if we can identify the virus. Okay, and then the one other thing I just want to talk about is I think that this connection in Egypt really offers the promise of many future studies for people at Duke or Maryland or wherever in terms of looking at hepatitis C. There are many aspects. I think that you could do any study related to future HCV treatments. I think particularly if you were looking at low-cost alternative or complementary agents, this is a place where the virus is rampant, but resources for the most expensive new agents are not there. So if you have a low-cost alternative or complementary agent, this is a place to do it. My colleague, Dr. Al-Kamari, he actually has a study on Silimarin, which is milk thistle, which has long been reputed to have anti-hepatitis C activity, or actually protects against many different liver diseases. And so he has a study that's funded, that's ongoing about Silimarin treatment in hepatitis. Certainly because of the interaction between hepatitis C and this parasite, schistosome, there are opportunities to study how they interact. It's known that schistosomal parasite really skews your immune responses. It creates a very fibrogenic response in tissue. So the parasite, when it infects tissues, creates this layers of fibrotic tissue. And certainly for a hepatitis C, where the main damage is fibrosis in the liver, having a fibrogenic environment is bad. And so the idea that these two sort of are synergistic in the damage they do to the liver is an aspect that I think is known but understudied. The other thing is detailed epidemiologic investigations regarding unappreciated horizontal transmission. So we know that the virus, even though it was inoculated in this age group that received the IV tartar medic, we know that the prevalence, that the disease has been self-sustaining, that it's found ways, aside from vertical transmission, to propagate in the population. And so perhaps, given the number of patients who were there, trying to understand some of these less prevalent methods of transmission, you could do those studies here, where you couldn't do them here. And then obviously new prevention strategies. If there are novel ways to try to prevent transmission of the virus, certainly you could do them there. Other HCV-associated morbidities or comorbidities. We've been involved in some studies here looking at gestational diabetes. And the link to diabetes overall, hepatitis C is known to be associated with diabetes. Given the number of patients there, you could do studies looking at that. And certainly any new low-cost testing methods you could do in a place like this that's relatively resource poor. It's not in the lowest rung of the economic ladder, but certainly it's sort of in the middle. And certainly some areas are less resourced than others. So I think there's a lot of opportunity, and what I hope is that this initial study that we're doing becomes an entryway for some of our colleagues here, perhaps some of you in the room, to really get involved in some of the studies that are there in Egypt. So I'm hoping that this sort of builds as a hub of future investigation. And that's really it. So shukran is thank you in Arabic. So that's the one Arabic word I learned when I was there. And I'd be happy to take any questions from you. Thanks for your attention.